Chapter 3, Basic Principles and Concepts, Immunology and Pathology, Adaptive Immune Responses of Vertebrates, Part 4. When we look at the immune response, we see that there is a series of reactions, a series of steps that will occur, that must occur for certain things to happen. We can split these events into certain camps, certain paths that we can see that the immune response will take. Path 1, Path 2, Path 3, Path 4. We're going to look at these 4 paths in this mini lecture. First pathway is going to be what is referred to as delayed type hypersensitivity. And if you notice right here, it talks about Th1 cells. Th1 cells, okay, remember this is going to be the class of CD4 T cells, T helper 1. T helper 1 cells, they're responsible for going and activating or hyperactivating, I should say, the innate immune cells. Th1 cells, once activated themselves, because an APC brings them an epitope, do you recognize this? MHC, T cell receptor interact. Turns out the epitope does bind to the T cell receptor. There's the co-stimulatory signals. Th1 cell now activated. Th1 cell will now go and start upregulating what we think of as inflammation. Inflammation meaning fluid retention in the area of the infection, the area of whatever organism's not you. Increase cytokine, chemokine production by cells that are there. Increase integrins and other cell surface markers that are going to start recruiting the innate cells, more innate cells to that area. And then ultimately, as you see there for that third sub-bullet, the macrophages. They're going to have certain genes turned on. These genes are going to increase their ability to bind, phagocytize, and then kill anything not you. They seem to have found that the schistosomiasis, that disease, the schistosomes, this is the type of immune reaction that they trigger more often than not. The second pathway we can see, the second route, is what is referred to as Cytotoxic T Lymphocytes Cd8. This is going to be what happens when you end up with, I'll say, like an organ transplant, where the surface markers are not close enough to match, so the immune system immediately recognizes it's not self. The activated CD8s are going to become these special subtype of cytotoxic T cells. They are going to go and they are going to start to kill those organs at the cellular level. They induce apoptosis or apoptosis, depending upon, where you're at in the world. Apoptosis. Apoptosis or apoptosis. Whether or not that second P is silent. Don't know, don't care. Your book keeps saying cell lysis, cell lysis. No, you do not want to have cell lysis. Cell lysis means pop, burst, cell death. Now, yes, that we want to see. Apoptosis, remember, the cell itself starts to, once that apoptosis is triggered, it will start to cut up its own DNA. Proteases get activated. All sorts of things start just chewing up everything from the inside. The plasma membrane stays intact, and the cell itself just kind of collapses, shrivels up, so that macrophages can now come and clear them out. We're going to see that this is the chief response to malarial diseases. Diseases intracellular bacteria, viruses, and intracellular parasites. Think of those most often. They're also going to get the natural killer cells. Okay, these are large granulocytes. They are what our immune system will trigger before CD8s. Okay, so with the CD8s, it's pretty much going from 0 to 60, in 2 seconds flat, which your body really sometimes just does not want to do that. So there's this little intermediate. So instead of 0 to 60, we're going to ease it up a bit. We're going to go up a few notches, and then if we need to, we can go further, or if this is enough, then we'll stop right there. That's what this natural killer cells are. Their name tells you exactly what they do. They go, they find, they kill. End of story. How they find, how they kill specifically, like I've said before, take immunology. So they're going to be the pre-CTL. They're going to be the buildup, the next step before you go all in. This is transplants and viral infections. Okay, you got to remember viral infections. Generally, the virus outside the cell, remember, it's inert, so it's not giving off any metabolic waste. It's not shedding any proteins and things like that. So for a lot of viruses, they can escape detection until they're inside one of your cells, inside a host cell. And then they will trigger different things to be on the surface. So instead of the normal proteins sticking up off the surface of one of your cells, the viral infected cells is going to stop expressing some, which is a signal that there's an issue with that cell, and some of them that they already are going to be altered. Another trigger, another signal that that cell is in distress, maybe virally infected. And then you get that whole MHC thing. Every so often, your cells will just chop up a random protein inside of you and present it to the surface. Remember, all the B cells and T cells have already been chosen to be ones that won't recognize anything inside you. So these proteins that, dendritic cells deliver, no B cells, no T cells will recognize it, all's good. Sometimes, if it's a virus-infected cell, one of those proteins that get chopped up would be a viral protein. It's a viral epitope. Dendritic cell delivers it, there's going to be a B cell or T cell that says, wait a minute, hold up. Th1CD cell goes, activates, natural killer goes, and that first one, the delayed hypersensitivity, activates a macrophage. Natural killer cell will go find that one cell that's virally infected, kill it, cause it to go through apoptosis, and then the macrophage comes in and wagga, 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 cleans up the mess. And then you have the immediate hypersensitivity. Okay? The delayed, as the name says, takes a bit, minutes, hours, maybe days, depending on whether or not this is first exposure, for you to mount an immune reaction. Immediate, it's going to be within minutes, hours at the most, but mostly within minutes. Because you already have the IgE, immunoglobulin E. It's right there at the mucous membranes. It's in close proximity to one of its target cells, mast cells, eosinophils. IgE, so antibody that's going to be found around mucous membranes, sinuses, eyes, whatnot. It's the one that we closely associate with allergies. For some reason, your IgE will start binding to things that you become exposed to, pollens, dust, dander. Triggers eosinophils, triggers mast cells, and somehow still triggers Th2 response, even though the Th2, the CD4s, are supposed to be a little more closely regulated. But because the IgE is binding, because the mast cells are dumping histamines and things like that, Th2 cells are going to just get caught up in the fray, meaning that the Th2 cells will then go and start activating eosinophils and macrophages and all sorts of things, and the inflammatory response in your sinuses, around your eyes, and places like that are going to go plump as inflammation hits and they swell up. And then you sound like this, even though you don't have a cold, but oh my God, I breathed in some dust. There you go. Originally, this was thought to be the parasite killer. Okay, IgE is mammalian only. You only find it in mammals. So they think that this IgE response, this cell-mediated immunity, this immediate hypersensitivity, may have been one of our evolutionarily chosen mechanisms for dealing with exposure to parasites. And as I've stated before in lecture, over the past century or so, we have become less parasite-carrying individuals, especially here in the West, the US, Canada, Western Europe. Hey, guess where allergies are going through the roof? Places where we don't have parasites. So it turns out these IgE, this immediate hypersensitivity that we've been evolving to have for the past few million years, got to have something to work against. So tree pollen, grass pollen, dog dander, things like that have now become the targets.